If we could ask everyone to take their seats, we're going to get started.
Hello, hello.
If we could get the panel back to the table. Okay.
Hello? Hello?
Okay, if we could, is this on? Okay. Yes.
Hello?
Okay. Could we ask everyone on the panel to please take their seats up front and we'll get started. Great. So I'm going to turn the gavel over to Dr. Nambiar, who is the Division Director for the Division of Anti-Infective Products. She trained at Children's Hospital in Pediatric Infectious Disease, and for quite some time actually was part of running a joint fellowship program we have in infectious disease between Children's and the FDA. And she took over as division director about 2 years ago now, I think. So thanks, Samadhi.
Thanks, John. So before we go into the actual panel discussion, I would like to extend my sincere thanks as well to patients and patient representatives for participating in this morning's discussion. Hearing from all of you about the issues that matter most to you are of tremendous value to us as we move forward in finding safe and effective treatment for this condition. So we've heard from experts in the field this afternoon who provided an overview of NTM lung infections and set the stage for the next discussion. And Dr. O'Donnell's talk has made my task a lot easier because I think a lot of the points I wanted to raise have already been brought up. So, we only have about 90 minutes to go through a lot of topics. So we'll try and focus on 4 key issues. The first 3 really relate to trial design. We'll talk about the appropriate trial population, the endpoints, and choice of appropriate controls for the trials. And then the 4th topic, which is just as important, is regarding practical considerations in conducting these trials. And it was briefly mentioned this morning, did not come up much in presentations this afternoon, but I think it's an important point to discuss as these trials are certainly of a long duration. Treatment regimens are very complicated and the frequency of treatment visits can be quite challenging. So with that, I thought the first topic that we can talk about would be the populations and how might we streamline our trials and who would be appropriate to enroll.
So they're going to have trouble hearing you, so let's move this over here.
Can you hear me better now? No.
Let's make that right up there.
Okay. No, this might be too close, but okay.
No, it's perfect.
All right. So in terms of the populations to study, and I think Dr. O'Donnell laid this all out very clearly, is it all right to limit it to only NTM patients who have underlying bronchiectasis? Is it okay to also include patients with cystic fibrosis in the same trial? If so, do we stratify them? We've heard in the discussions today that these two populations can be fairly different in terms of their outcomes. The frequency of the pathogens might be different between these two sets of patients. And there might be differences in other risk factors such as steroid use or macrolide use as well. So that would be one question that we would really appreciate the input from the panel. The second key area would be differentiating between those who are treatment experienced versus treatment naive. Is it okay to combine them again in one study, or should they be studied separately? Potentially one can have different trial designs depending on whether it's a naive population or an experienced population. Would we consider different endpoints, again, if they're treatment naive versus treatment experienced? What might be the treatment benefit? Would there be a difference if this is a treatment experienced patient versus being a treatment naive patient? Again, Dr. O'Donnell has addressed this, whether MAC patients and non-MAC patients should be in the same study. If so, should they be stratified? Are they different enough that they should be kept separately? Patients with nodular disease versus patients with fibrocavitary disease. So I think the bottom line is we've heard today is this is a very, very heterogeneous disease, and what might be the best way to study it in terms of the patient population. So I'll pause here. I would welcome input from the panel members, and then we'll move on to the next topic. Any takers?
Okay, so I'm going to answer all of these. No, just kidding. First of all, thank you to the FDA for convening this incredibly important meeting. I'll just start with that because this is extremely important for our patients, for us as clinicians. Thank everyone who's here for their input. So, I mean, I'm just going to run down that list real quick. CF versus non-CF. So I believe that this is a need for both populations. It is a significant health issue for both populations. And yes, they're different, but in my mind, they should be included in trials. If you marshal the resources to put on one of these trials, why would you exclude a population of need? If you recall, we did that with HIV and TB for way too long. And now all TB trials include HIV as well. Yes, they're different. We can deal with that in the design and the analysis. So I think, yes. Naive versus experienced. This is a practical issue. Most of the naive people, or the ones who haven't been treated, are out in primary care offices and pulmonary offices. They're very hard to find. So from a practical standpoint, you know, if you really want to get a lot of patients, they're going to be coming to the reference centers that we keep here getting mentioned today. And I just think from a practical standpoint, leaving them out is really problematic in terms of being able to find patients to treat. MAC versus non-MAC, that depends on the MIC. That decision, science should just drive whether you include MAC and obsessives. Not all drugs that work for one work for the other. I think if they work for both, same argument as to me as with the CF question. They should both be included if the drug works for both. The most important thing, and this really wasn't, I don't think, highlighted well enough, is that really what matters is the precise speciation of the organism. Anne mentioned this issue, but really it's about speciation, and that means it needs to go to the right laboratory so that it can be speciated. Or how do you interpret the outcome of someone who's treated with abscessus versus Massiliense, as Anne said, where one does really well and the other doesn't, but most labs don't distinguish the two. The final thing here is there's so many confounders here that I want to propose this idea, and I want to hear from industry. They won't like it. We can't tell who's going to progress on day one. It's not possible. But we do have a little better idea at about 6 months. So in South Korea, and they published this approach, when they see someone, they follow them for 6 months, collect additional sputum, re-image at 6 months, and now at least they've had 6 months to say, this is a progressor. This patient's not a progressor. And I think it helps you find the right people to be enrolled in a trial. Otherwise, at 6 months, many of these are going to be stable. And those are not the people you want to enroll in a trial. You want to enroll the progressors to be able to understand, did the treatment have an impact on any of the outcomes which we'll subsequently measure?
Are we done with that question?
No. That is just the beginning.
Okay, that's just the start. Well,
Yeah.
so, we were just in San Diego, some of us. I was there less time. I'm sure I'll hear about it. But we spent 5 hours talking about just one question for the new guidelines meeting. So maybe we should move to the next question. But I want to echo Chuck's comments and thank everyone for doing this and for putting this disease on the map. I think it's the last few years we've gained a lot of momentum, and a lot of that has to do with the patients here and the patient advocacy groups, as well as the FDA and industry and the academics, and we're all in this together, and we're all trying to accomplish the same thing. So I'm very happy to see movement in the last few years. I liked all of Chuck's answers, so I'm not going to weigh in and disagree. And in fact, we just submitted a grant today that took the approach that Chuck just mentioned. I guess we're going to get to the comparison groups later, but I think the idea that everyone needs to be treated right away is obviously not true, and we know that, and it's in our guidelines that we support watching people who clinically can be watched, and at some point I feel that most people do need to be treated or they'll want to be treated, and exactly when that happens, of course, is a spectrum and it's a case-by-case thing. It's different for everyone. So I do think that the answers Chuck provided, I would just say I'd agree with them. And I don't like excluding groups up front. I'm an epidemiologist. I like to handle it in the analysis. And as long as you can construct a big enough trial, and I know this gets challenging because it becomes more costly, but it's really silly to exclude people up front if you don't have to. And it's better to sort out who responded later in your analysis and looking at subgroups and things like that. So I think as much as possible, we should try to be inclusive.
I'd like to make a comment that it depends on whether the trial is primarily a safety, that is, the drug's never been used in any of these populations, or as we are with their case, we've already been through one trial. Now you're probably more interested in efficacy, and safety is much less of an issue. So if it's a safety issue, the more patients you put in the trial, the more you may be able to answer that question. But an efficacy trial, it may also be helpful to put them all together if you can divide them all out. I talked earlier about the design trial of having patients come every month and dealing with a patient population that averages 75 years of age. It is not a possible trial. We lost so many patients. We did a trial just with inhaled amikacin and got 2/3 of the patients because they were unable to find a way to get there to do that. So if there's anything to emphasize, it would be that. So some difference based on whether it's safety or efficacy, and that would, I guess, be whether this is the first trial or the second trial. I agree with Chuck about CF. As long as we're going to do them, we can always split them out. The clinics are often together, fairly close together. I mean, I think we could be able to do that. I think M. abscessus, as Chuck alluded to, the difference between the isolates that are macrolide susceptible and macrolide resistant on the basis of the ERM gene is enormous. Obviously, there's only been maybe one trial, but none of us were surprised by the results. Again, I don't know if you have to separate them out, but you have to identify them. We have to know which ones are macrolide susceptible, which ones are masseal lens, so you can separate them out when you get there. Including, I think, with the recently completed ARICASE trial that had both abscessus and MAC for safety reasons, I think that was fine. Now, if you want to do an efficacy trial, you might not make them the same. I don't know we've thought about that in terms of what I mean, the next trial is going to look at just one of them, but they may not be treated the same. They may not respond the same. I actually don't know that. I mean, in the sense that you had one group that responded better than the other, but I don't know if it was study design. Should you have gone longer before you assessed them or not? 6 months isn't very long for M. abscessus. So I pretty much agree with Chuck. The one other thought is that if the endpoint is primarily or exclusively microbiologic, it doesn't really matter to me how sick the patient is. The endpoint is fairly objective. You can either make the cultures negative or I mean, they need to be treated or you decide, but I wouldn't, 99% of our patients come to us because they're symptomatic. Somebody has referred them to us, primary care, pulmonologist, or why did they have the test done in the first place? They had some kind of symptom, so most of them are usually willing to enter the trial at that point. So I don't know about the 6 months if the endpoint is primarily microbiologic. Patient-reported outcomes, it is tougher if the patient doesn't have a lot of symptoms. I mean, if that's very important, then you may want to separate them out so that the ones that are the sickest, you can really use patient-reported outcomes as an outcome as compared to some patients who they don't feel well, but they're not going to give you they're not really sick in the way that you could probably measure that very objectively.
So I would just like to add that, like, when you're actually sitting down to put these clinical trial designs together, you think you've got it, and then you pressure test it, and you wonder whether you are doing the right thing when you're going to put so many patients through the trial and resources. So one of the things I want to highlight that hasn't actually been spoken about is the timing of the endpoint. Not just the endpoint. Right now we have the microbiological endpoint, which is the gold standard, whether that is, the right thing. It's clearly a biomarker, a surrogate, and we do need, correlated patient endpoints, whether they're functional or whether they're patient-reported outcomes. And we would welcome a patient, a PRO for these trials, because it seems like if it's one that's properly validated and works, it will solve a lot of our problems. We don't have it at this moment, unfortunately. But when we looked at, in our experience, when we looked at our trial, we did have, non-CF, and we had CF patients, and we had abscessus, and we had MAC. And when you look at, what happened, they behave differently. And so when we're trying to put another trial together, we do want to include all the patients. And the question for us became, do we include them all in the one trial, or do we actually have separate trials for them? Because we believe that if you believe that all of these patients could benefit from your drug, then you have to actually, do clinical trials and include all the patients. So the question isn't, do we need to do clinical trials in all the patients? The question is, how do we design a trial so that we capture, whether the drug works or not? And when we looked at our trial, the non-CF MAC population appeared to respond in terms of their cultures much, much faster. And the clinical trial wasn't long. It was 3 months double-blind and then another 3 months open label. And in that time, if you really dig down and look at it, I can say that some of the M. abscessus patients look like if we gave them enough time, we may have seen something, but we didn't. And so the subpopulation that really stuck out and seemed to do really well was the non-CF MAC population. And when you're pioneering trials in this area like we are without there's a paucity of prospective controlled clinical trials. It's very difficult to draw your assumptions, and without assumptions, you're literally just stabbing in the dark. And so, we have some assumptions, and that's why the current study that we're doing is a much narrower population. And we'd love to do, and more studies, and hopefully have the PROs ready to go when that happens. And then, and also in our experience with the narrow, the narrower, the non-CF MAC, we didn't know what outcomes, we were going to expect that were going to actually show something. And, it happened to be that the 6-minute walk test that was in this trial. And we don't really know what that means at this moment, other than we do have, a separation. We did see a separation in the non-CF MAC patients. They seem to do much better. And furthermore, we actually saw, when we actually looked at the data even more, that the converters amongst that subgroup drove that result. And that's what we're left with now to actually, put it in the next trial, and we hope to see reproduce, that result to be reproduced. But that's really the difficulty, is the timing of the endpoint, because you can cut the data. I mean, you can analyze it, you can stratify patients, and you can analyze it a number of different ways. But if your endpoint is at 6 months and your MFSS patients are going to convert or improve in their PRO at 8 months or 9 months, you've missed it. And that's the danger, and that's what we were afraid to allow it to happen.
I would just bring up the issue of numbers and practicality of accrual. So I think we're very early on in development of clinical trials from this, and if we get too narrow and too focused too early on, we're not going to get clues about how these different subpopulations may or may not respond to a drug. The tremendous successes that cystic fibrosis has had in terms of clinical trials, I think in large part is related to the easy accessibility of those patients, with them aggregated into clinical care centers, and the willingness of the patients to participate. But that comes on the backs of what we see now of years of building excitement in that disease. We don't have all of the patients aggregated into geographically dispersed centers throughout the U.S. And again, we're very early on in development of trials, and so trying to get that excitement to build, trying to get patients encouraged to participate to where we can get the types of timely accrual that we need, I think is an issue. And, when you're looking at things like MAC versus M. abscessus, there may be differences in, right or wrong, the perceived need of new drugs. And I think many of us would agree that M. abscessus has a pressing need for something more effective to develop. When we actually sit down and look at the numbers that we have access to, if you say that these organisms are more important in the setting of cystic fibrosis, we can calculate fairly quickly, and I think fairly accurately, how many CF patients in the U.S. have these infections, and it's not very many. And so if you were going to try to design a trial of a drug to look at M. abscessus in cystic fibrosis, I think even in that disease where it's relatively easy to accrue, you're going to have problems. Whereas MAC, I think that there may be more patients, especially non-CF patients, that have it. Finding those patients and getting them to participate may be a problem. When it comes to treatment naive versus treatment experienced, I think it depends, again, the sort of stage that you're developing the drug in and what you're looking to show. If you're trying to introduce a novel drug into treatment naive MAC patients as part of a multi-drug regimen, as David and Richard have discussed, the proportion of patients that will respond to the standard treatment is pretty high, and it's going to be difficult to get numbers that will show a benefit of adding or substituting a new drug in for those patients. On the other hand, if you take a patient population like we started with the Insmed trial, that we had some documentation of their refractoriness to their sort of optimized background regimen, I think from a number standpoint, it becomes easier to introduce a new drug, and if that drug has effectiveness, to be able to see that in the context of what people would consider standard of care. So we'll talk about study designs next, and there may be other ways of getting around that, but I think those are considerations that need to be kept in mind when you're talking about the dichotomies that we're discussing in this section.
Yeah, if I could. So, so again, as the others have said, thank you to the FDA for convening this. This is a critical need. Great to have the input, particularly that we had this morning from patients. So I'd like to, so Chuck, yes, I like your idea, actually, because we have to find some way of trying to predict or enrich the population and somehow to identify those who are likely to progress and that you can demonstrate a difference. Because I think the challenge we will have is probably from my statistician friend, which is how do we handle then that data set, because we'll draw the line on time 0 and it will be finished 30 months later. And we have to, figure out how between that it's going to take some creativity to, I believe, and really some novel thinking to find ways of assessing particular endpoints in ways that will allow a drug to be approved to the point where we believe there's a good benefit risk of that product. And it's really quite a challenge. Also, that last week in San Diego, call from the podium, which is we're having to do the wrong trials in the wrong populations. Right? The path to registration for new antibiotics now is in skin infections or UTI. And it's not the challenge that we have, but because we have that path there, we're allowed to follow it because we know how to design the trials and select the populations because we've learned from that. And now we're sort of having to learn the difficult way of complex patients, very heterogeneous rate of progression. And that really does challenge us. So I think hopefully in the next little while, there'll actually be some more flexibility in terms of design for an area where there's such a big need. And being a little bit more flexible up front in terms of what has to be pre-specified, because otherwise the number of patients and the overall cost, the amount of microbiology testing you have to do really is a barrier for good studies.
I would like to just mildly dissent on a couple of points. In terms of the 6-month waiting period, there are, very clearly people who are symptomatic and have progressive disease at the time of their initial evaluation. And I think that there would be no benefit gained by asking those people to wait further for initiation of therapy. As a matter of fact, I think they'd go somewhere else. But, I mean, the routine for me is at least half of the people I see, I don't start on therapy at the initial visit, but within 2 or 3 months, I've got a pretty good idea about whether or not they need therapy. But I just think 6 months is kind of rigid and a little bit long. Perhaps it could be, maybe there's room to compromise in that. And the point, though, about MAC and abscesses, they're totally different. And if you go into it knowing that you're going to post hoc separate the analysis, why wouldn't you do it initially? I I understand that you have to have numbers, and that's really a lot of what we've been discussing is patient accrual, and clearly we're looking at multi-center trials in anything that you talk about. You've got to marshal all of the resources in the country to get the power to do statistical analysis, but there's no overlap in medicines between MAC and abscesses, except perhaps the study medicine, which like Eric. So, I personally, I think the way that INSMIT has approached the second trial makes sense, but I wish they would do another trial for abscesses. I just don't. I'm all for treating them all. Why not include simiae? Simiae is such a difficult bug to treat. Why don't we try a few simiae patients? Well, you're not going to lump them with MAC. At any rate, I'm sorry, that's just my thought on that. You know that you're going to separate them in your analysis regardless.
I just want to address the getting more patients into trials and spreading out our network, because clearly from hearing from everybody today, that sort of makes me feel sad for our profession that, patients aren't getting diagnosed in a timely fashion. So, one thing we're trying to promote is this bronchiectasis research registry, which has an NTM component to it to try to get more patients involved than actually a patient-powered network, so patients themselves can register to potentially be in clinical trials. So that's, we've got to get this word out better. We're not doing a very good job, I would say, in getting patients, patients in the room, great, but there's tons of other patients out there that we're not getting into clinical trials. The second thing I'd like to pick on a little bit is this treatment-naive patients, because maybe we do need a trial in treatment-naive patients. My clinical experience, and I bet many patients will say rifampin is the hard drug for many patients in the current regimen, and maybe we need a trial that looks at something other than those three and substitute something else for rifampin in a head-to-head trial. I know Dave is giving me the evil eye there, but I do think we don't have the final answer for treatment-naive patients either.
Well, we discussed it briefly at the meeting. Where do you put your resources? What's the priority of your questions? And that's tough. Is that a question you want to take a sizable number of people to enroll in a trial to answer when they might be in another trial?
Yeah.
It's a tough call.
Yeah. So first of all, I want to just reiterate that we actually do believe all of these patients need to be studied, and we're committed to that. And just with respect to the treatment-naive patients, I'm wondering if this is a time to talk about separating that group into, the cavitary lesions versus not having cavitary lesions. And the reason for that is because without cavitary lesions, the current standard of care has a high, success rate, which makes, the sample size that you need for your trial in this disease, almost impossible to achieve in a reasonable period of time.
Just a few comments from somebody who sees development in a bunch of different drug areas, but doesn't know very much about NTM. Just to underscore what Dr. O'Donnell said about, it seemed to me that in the CF world, that having that registry data is very, very helpful to trial designers in terms of understanding the optimal timing of endpoints with patients with certain characteristics, et cetera. So just to sort of underscore that's a very important effort, I think, based on my own observation. It really helped them move forward. I think one of the things just to point out is that if the patient population is heterogeneous enough that there are groups of patients where one expects the optimal timing of the endpoint to be better, this is not an FDA opinion, it's just my personal opinion, that one might want to consider two trials with different patient populations with different timings of endpoints. And Karen Higgins, I invited on the panel, make sure I don't say something heretical. She's from the Office of Biostatistics. But I would think that if both of those trials were successful, they could support each other, and you then have a package of substantial efficacy. And I know that funding is limited, et cetera, but that's something that has been effective in other infectious disease areas as far as getting to approval. So.
Just very briefly, this could also be done, these two trials, kind of under an umbrella protocol where the two trials are considered separate, but for logistical reasons it would be easier to potentially enroll patients and put them in the trial that's most best for them.
Can I just comment, though? We don't have enough patients, to do that, and the need is so great. And you know how many examples in medicine where that's what the company said they were going to do, and they didn't do it. And it's left to the clinician to work off-label with no data. So when we have a chance to enroll patients who have a great need, we need to rethink that, I think. And it may not be perfect, but I still think it's a really important issue that we go and we study non-CF because there's more of them, and then we just don't study CF because it's approved and everyone says now, CF Foundation, you go pay for that, or academia, you pay for that. I think that they're all sitting in the same clinics, usually in the same hospitals, so there's a real strong argument, mainly based on need, to get them studied early, not later. The 6 months is an example of trying to use some kind of marker of progression. It could be you use the previous 6 months, too, to be able to identify progression. That's really all all I was saying. And one thing that I think is another issue that's really important, whether you decide to do a separate trial for abscessus or MAC, recognize that in our study of abscessus, 55% of the patients also had MAC concurrently or previously. So this is a problem in terms of trying to figure out how to design a study to look for efficacy when there's so much recurrence and reinfection.
And also add a lot of them had Pseudomonas, and they have Aspergillus, and they have Stenotrophomonas, and everything else growing in there. So it does get complicated.
So I think we've heard quite different viewpoints on this issue, so it hasn't made our life any easier, but I can see the merits of doing a study which is sort of in an all-comer population, so you haven't really restricted it to one subgroup versus the other. I think that makes a lot of sense in terms of extrapolating to the larger population. But from a practical standpoint, I think it does pose some challenges. So Dr. Wallace, I think you had a question earlier about whether the study was being done for safety or efficacy. I think the discussion here primarily is about assessing the efficacy and really deciding if a new therapy works or not. So our focus here was more in the context of an efficacy trial, but I think your point is valid. That was less of an, not that it's not important, but becomes less of an issue with safety studies. And we will get some safety, obviously, from these efficacy trials, but sometimes if the numbers end up being small, the treatment effects large, the sample size may be smaller, then we might ask for a separate study primarily focused on safety. We'll collect some efficacy data, but it's not powered for any kind of efficacy assessment.
Can I just add one thing?
Sure.
The only thing I'll add is that I think it depends on what the question is a bit, right? And we're all kind of talking about the same thing, but no one's articulating the question. And if the question is, does the drug kill NTM, that's a different question than does the drug make CF better? Does the drug make non-CF bronchiectasis better? These are different questions, and so you are going to potentially use different measurements of efficacy for those individual groups. You might study them different lengths, and so that that's why we are tempted to segregate. But again, if your question is, does the drug kill Mycobacterium? It might kill it to a different extent or over different time periods in different subgroups of people. But if your ultimate question is, does the drug kill Mycobacterium in a human body, you can lump all those groups together. And again, I'm just saying it depends on what the question is. And I know we're getting to that next here.
Right.
What is the endpoint that we're actually trying to ascertain? So.
Right. I think the basic question is does it make the patient feel better? I think that's what we're trying to get to. And along the way, hopefully it's also killing the NTM and then making the patient better. So it's more than just killing the organism. I think that's what we're trying to find out.
So then the next question is killing the organism a surrogate for actually feeling better and living longer?
Right. So I think
and that's really what we care about.
Right. So that's sort of the next question. So maybe that's a good way to start the discussion on the endpoint. I think Dr.
I guess
John
that's a good question for Alexandra. How can we correlate the PRO with organism killing? Or can we?
Well, you
Or
definitely
the semi-quantitative cultures or something. Can we
You definitely can correlate them, and in the CFQR studies I've worked on, we did that. So we would correlate change in respiratory symptoms with changes in microbiology. And the FDA, of course, isn't interested in the microbiology as the primary, but it definitely correlated. So with greater killing, we saw better symptom improvement. And you can also correlate it with the 6-minute walk. We've done those kinds of studies, and yes, if you have a good respiratory or symptom scale, you can do that.
I can't think of another disease in which we briefly talked about it, in which you have two diseases active on the same part of the body, producing the same symptoms, and then you're trying to do a PRO. I mean, these are people with bronchiectasis, even if they don't have another identified tough pathogen, and you have an NTM, both functional, both inflammatory, and going through the symptoms. If your bronchiectasis stays calm, the PRO for the NTM will probably be great. But if they're having lots of trouble, they grow staph several times during the middle of this stuff. And it may count, if you count it negatively against them because they didn't show a response when in fact the problem was the bronchiectasis. It's such an unusual circumstance. We just have to think about the PRO and how to make it work when you have two diseases that are that are active at the same time.
I agree with Rich, and that's why I think killing NTM may be a surrogate for better living for some of these people, but it may not be for other people. Not that it might be harmful, but it just may not have the same effect.
I mean, there are people who come in after you've eradicated their MAC and say at the end of the treatment course and say they feel worse. And that's because they have another disease going on at the same time. Now, that doesn't mean that if we did enough numbers and whatnot that overall that they would look better, but.
Well, I think you're raising just a really amazingly great point. And one thing I've been thinking about this whole day is one of the advantages that we actually might have, and we would need to talk to our statisticians and things like that, but we do have a very well-validated respiratory symptom scale that's particular to CF and has proven efficacy, as well as the respiratory scale on the QLB. They're different. They actually reflect the different respiratory symptoms of bronchiectasis versus CF. So one thing that would be really interesting and perhaps an answer to this problem is to be examining both the respiratory symptom scales, which differ by disease, and the NTM module, which focuses in much more on what Kevin is saying. And that might enable us to see if the respiratory symptoms are going up, that might be a sense that, they colonize with Pseudomonas. So that might be one way that we could actually get around that problem. And I agree with you, it's complex.
I would just add, in our paper on the semi-quantitative sputum cultures, Kevin, the people who converted within 6 months, half of them had a bronchiectatic exacerbation during that 6-month period, and for the people who took longer than 6 months, it was almost 80 or 90%. So there's no question that the bronchiectatic symptoms are going to complicate the deal. If you can separate them out, that would really, really be helpful.
It's a cheap test. We just have to fill out some forms and paperwork, and it'd be very informative. We haven't done it, and we're assuming that it won't help, but shoot, I'd rather do it than not do it. These are great patients. There are only a few things that keep them from saying they always want to be in trials. Virtually every one of them, and it's only characteristics of travel or a little bit of reservations about being on the placebo side that keep them from being involved. So, I bet they'd fill out those forms in a second.
I think another complication of is the toxicity of the drug and how that complicates how the patient feels. So if you just take the example of an inhaled drug that might have airway toxicity, that if the mycobacteria is killed, it may eventually make them feel better. It may increase their functional status. It may make them live longer in the long run. But during the period that they're inhaling the drug, they may cough more, they may wheeze more, they may feel more short of breath. And it becomes very difficult to tease out exactly what's causing that PRO when you throw that complicating factor into it as well. And it can be any toxicity for any drug that might affect that particular part of the PRO.
I agree. It's going to be hard to discern disease-related symptoms versus treatment-related symptoms, and that might be another module that needs to be added into a questionnaire to maybe try to get to that and tease that out.
Yeah, that's a good point. And just to bring up, because I know you've been talking a lot about how these patients are complicated. They have a variety of risk factors. They're somewhat heterogeneous. But remember that randomization is a wonderful thing. And that if it works, the risk factors should be relatively even between the arms. I think one of the things related to the Chuck, we'll talk about the Chuck approach, is that it points out to me that if you are going to have a very heterogeneous trial, it might be important to stratify at enrollment so that you make sure that you've got your risk factors balanced between arms.
This brings up another reason to follow the Chuck approach. So one of the things that we've noted at National Jewish, and I'm sure all my colleagues have, is that if someone comes in with bronchiectasis and mycobacterial infection, if we introduce airway clearance, their cough will improve within 2 weeks, 3 weeks, a month. And so if this is a multi-center study, if that varies across centers, how aggressive you do airway clearance, which approach you use, do you prevent reflux from occurring, then that randomization may get screwed up because that you can't randomize practice. So if you had a period where you started a certain protocol, like a run-in airway clearance, check that PRO instrument to see how much better the symptoms got with that intervention, before you started the antibiotics, it would clean it up. That's all I can tell you. It would clean up the ability to understand how symptoms change with antibiotic therapy.
Could you just try to standardize the pulmonary care that you gave them across? I mean, it clearly means something that attention should be paid to it, and it should not be left to just each institution to do it the same way. I mean, the only way it would work is now admittedly, respiratory therapists are not the same, but you're making an effort to know they're at least they're all using saline, they're all using, whatever type of equipment and stuff you want to use.
Well, but for trying to look at the efficacy of an intervention, an antibiotic, and you want to know what that antibiotic contributes, or that regimen, you can't do what you just said.
That's true. We don't even, maybe Ann can correct me, but I mean, when it comes time to tell these patients what to do to treat their bronchiectasis and what makes them live longer and do better, we don't know the answer to that question either.
I mean, I think, Dr. Eagles, I was wondering if in your experience, how you've been able to handle the other therapy.
So one of the things that I was going to mention is that this is a rare disease, and we don't have that many patients, and so when you start to put your pivotal trials together and you need to power them up with a sizable number of patients, you're reaching out to a large number of sites. And we already heard today a number of times that, once you start moving away from the experts in this room, it becomes very difficult to have the same type of care. So you need to recruit your patients in a reasonable amount of time, and these are the real challenges that we have, and they're very real. And so
when the room was surveyed, only 57% of people were using airway clearance in this room. So, but I think what Chuck proposes is possible, like a 2-week run and just use a device, not anything more complicated than that. That could be done. I think hypertonic saline and all that kind of thing is more complicated.
I was going to bring up one more thing, just as a difficulty, because of the toxicities of the medications during the treatment. And right now, following the guidelines, while a patient has a negative sputum, you still need to continue treating them. And it's for a long time. It's 12 months after you have that first negative culture. And so the best time is probably to look at a PRO or any other measure of function, or once you have durability of that treatment success. But the challenge is, you're facing a trial that has an outcome like years from when you start. And in a disease area where, you need to, maybe fast track whether something is working or not, it becomes very challenging to do that. Although, in our trial, we are looking at these, the PR, we're looking at function and we're looking at the sputum 3 months out from treatment success for that very reason.
Dr. Quitner, would you have any thoughts on what might be the right time to use the PRO to make a final assessment of the patient's getting better or not? I think, as Dr. Eagle said, usually treatment is long. And do you do this on treatment? Do you take a break after treatment to make sure things have gotten better? Welcome your thoughts on that.
I mean, this is a very important point. I can tell you for antibiotics and cystic fibrosis quite well because I've worked on so many clinical trials in that area. And so one of the really important things we did in those studies, which I don't think would work here, the run-in I think is a good idea, and measure the PRO, is that I told the various studies to be sure to measure, of course, baseline, but 2 weeks into the antibiotic, because we were fairly certain that it would have a lot of efficacy at a month. But these were shorter studies, obviously. These were in the days when the CF studies were much shorter. But I told them measure it 2 weeks because my sense of doing other smaller studies was that the efficacy was really there at 2 weeks. So we actually did baseline 2 weeks. In every study that was approved and successful, they saw the improvement at 2 weeks. And then we measured it again in a month. And then if they were going to take them off the drug, we waited a month later. This is obviously a different paradigm where it takes a lot longer, but what Chuck was suggesting was sort of a baseline run-in, 2-week or 1-month point, and then where is it that you really think these drugs, which we haven't talked about which ones, where do they have their maximum efficacy? Is that 6 months? Is it a year? And you measure it at that point as well. And then if you're going to take them off or you're going to wait, you have a follow-on. So what would that endpoint be? Where would you all, the practitioners, say is what you believe you get efficacy at that point?
Don't look at me.
That takes us right into the timing of the endpoint. So maybe before we go into timing, I was wondering if there are any more comments on the microbiologic endpoint, any other thoughts from the panel on the merits, demerits, the timing of the endpoint, whether it's 1 culture, 2 cultures, 3 cultures, what the interval must be between these various sputum cultures, and also the merits of doing a culture a certain time point after treatment has stopped and how long that might be, because 3 months has been chosen, it's fairly arbitrary. Is there any science behind it? And would welcome any discussion on that topic as well.
Let me divert a little bit. I've listened to Ellen and I discuss semi-quantitative sputum cultures and its dilemma. There still may be some in-between points that you might be able to analyze. For example, positive on solid and broth media versus positive in broth only. In a way, it's semi-quantitative, but not as we discussed. We find it useful because we usually can measure what's happening to the patient, although we can't tell when they're going to convert or whatnot. But maybe that would be some sort of semi-quantitative. It's better than broth alone, which I found very distasteful because you have such a huge range of potential possibilities in terms of the number of the organisms that are growing. You could be 4+ and still be 4+, or you could be 4+ down to 5 colonies, which is an enormous response, and the report would still be the same, positive in broth.
Are there any practical considerations in terms of all labs being able to do this kind of semi-quantitative testing? Because I understand that your group is very comfortable or has been doing it, but I don't get the sense that that's
all of them do agar in broth. Virtually all of them culture it on agar media and broth. That's the recommended standard in the U.S. Now, for the European studies, do they do it in agar and broth too? Or? So you couldn't get counts, but you could at least report it. That's our main issue is in Europe where they do not. I guess in Australia too, they don't do counts. So
they culture it in the 2 media, but they don't use the scale and define the steps the way that you have defined them. That's the only difference.
And actually, many labs. This is very interesting. Virtually all labs do it, but virtually all labs don't report it. It's a check for contamination. It's a check for mixed cultures. So it has its values, but no one's recommended reporting it, so they don't do it. So it's there. You just have to ask for it.
Yeah. Other than semi-quantitative cultures, have you looked at times of positivity in broth?
I don't know that we've done it in the studies, but I do it all the time. Because if you have just one species there, MAC or a rapid grower, you can pretty well predict based on the cultures. For example, almost all MAC in the broth media we use are positive within 14 days. Almost all the rapid growers are positive in 7 days. So if MAC takes 3 weeks to turn positive, there's hardly anything in there. And you can bet money that the agar is going to be negative because you only have a few colonies that are in there. So but I don't know that we all use the same broth system, and I don't know whether you could actually relate one center to another. But I'm guessing that there would be some relationship, and that is an objective number that you could use.
Aren't there issues, though, with the quality of the specimen and how it's collected? And there's just so many foibles and sputum.
Well, I think along those lines, I think one thing to keep in mind is sampling variability, and there are a couple of issues on that. I think the confidence you can have in one negative culture is a lot less than you would have in, say, 3 consecutive negative cultures, and that's why I think the sort of terminology, even though it may not be definitively defined in terms of culture conversion, is a bit more attractive. The other thing to kind of get around that is the collection of multiple specimens, and that actually ended up working quite well in the INSMED trial, and that for each time point we had at least 2, and in most cases 3 cultures that were obtained either at the clinic or from home collection that allowed us to sort of compare the consistency of whatever finding we were having at that time point. So I think when we're building these studies, having some redundancy in that and having more confidence in whatever culture-based endpoint we're determining, I think was very helpful.
I was going to say, in National Jewish, we do the same as Richard's lab in terms of the semi-quantitative. The issues here are just so basic. It's sample collection, but it's also processing, too, that's very important. A lot of these patients have co-pathogens and require significant decontamination of the specimen. That greatly reduces the colony-forming units of the NTM, and that's going to vary. And that's why it actually is different than TB in this regard. And so where in TB we have really quite a lot of good data about the consistency of CFU over time, it's not that way in NTM. And it does, at least it doesn't appear to be. So even though it sounds good, I think there's some really basic standardization that would have to occur across labs to get to the same place that TB is in terms of being able to use these cultures and try to quantify or semi-quantify the counts over time.
We also don't know how it correlates with the extent of disease or...
Right, so that's the next here.
Yeah. So it's kind of like either we're going to have to go with a positive, negative, and then define the number. Because we really don't have a correlation between quantitative cultures and disease state, I don't think. Maybe Dave and Richard did.
I was going to say, we did correlate, they did predict sputum conversion, the improvement in semi-quantitative sputum scores. They also correlated with symptomatic improvement. Now, it was gross. We're not, yes, we looked at better or worse, but there was a good correlation in semi-quantitative culture improvement and symptom improvement.
And I was just going to say I applaud them for doing this because I think that study was extremely important to be able to do that. Now the issue is, can it be done across multiple laboratories? Can they all get the same kind of results?
I think the other issue is the species you're dealing with for reasons that you raised for M. abscessus and the susceptibility of the decontamination process. Having consistency in that semi-quantitation may be very different than what you see in MAC.
We're just reminding ourselves that on Australia, Europe, and the U.S., for the current trial, only has a single central lab. So you don't have the issues of smaller labs, all of which are very experienced long-term doing these. And so you would be doing the best you could to minimize those issues that might occur if you use smaller labs or less experienced labs.
So it certainly sounds like there are some issues purely from a microbiology standpoint. Some of them we could potentially address, but some of them we just have to live with and see how to work with the information available. But I think the bigger and an important question is, I think, what Dr. Winthrop had brought up earlier is how does a micro endpoint at the end of the day translate in any kind of clinical outcome, whether it be a semi-quantitative, and it's encouraging that we at least have data from one institution, whether that will hold true in other institutions, in other settings, it remains to be seen. But if we do use microbiologic endpoint as a surrogate, we have to be at least comfortable to some degree that it reliably predicts clinical benefit. I mean, that's the basic definition of a surrogate endpoint, as Dr. Shamsuddin had outlined in her presentation. So I would welcome your thoughts on how you think there is correlation, whether we define it based on sputum culture conversion or whether it's a semi-quantitative measure of saying the bacterial load had decreased. But if it does translate into clinical benefit, what might that clinical benefit look like? Is it a mortality benefit? Is it a symptomatic benefit? I think that's a big question we have to deal with in terms of deciding whether it's a primary endpoint, an exploratory endpoint, or a secondary endpoint. So we welcome your thoughts on that topic.
Med trial, but don't you have culture and QLB?
So one of the things that I would say just as the first statement would be it depends on the population and where that population is in terms of the progression of the disease. If you're, if somebody, has severely progressive disease with very limited functional capacity, I'm not sure eradication of the organism is going to, make that much difference to that. Whereas your, naive patient who's just, recently treated may actually get the most benefit from a PRO or from a functional. So I think, I would put that out there to everybody to address that question. And with respect to our experience in our trial, we actually enrolled a population of patients that had refractory, disease in that they weren't responding to standard of care treatment. And many of those patients, up to half of them, had more than 2 years, much like the patients here, long periods of treatment. And amongst those patients, the majority had fibrocavitary disease, or at least the presence of cavitary lesions. So we were looking at a specific population. So in terms of separating out the things that I'm talking about, we were looking at one end of that spectrum.
QLB and the cultures at the same
time? We did do the QLB, and we did not see anything significant, and it may just have been a function of the time in which we were looking at that endpoint. There was some sort of trend within the patients who received LAI for like activities and things like that, but I really wouldn't draw any conclusions. So we didn't have anything that we saw on these measures, we couldn't correlate that with anything as a result. The study was, it was a short study in terms of the double-blind phase where you would like to assess the difference between active and placebo add-on.
This is an area where, despite Anne's dislike of the 6-minute walk, I think having some functional assessment in there, I think is very important. It can be very difficult to tease out, especially in the short term, how the drug may make you feel better. But if if you can show that the drug increases your functional ability, that may have meaning that eventually the symptoms would catch up from that. And again, it depends a lot on the toxicity of the drug.
Mm. And because we did see a difference in the 6-minute walk test, we could correlate it, and it did correlate with the microbiological outcomes.
Microbiological endpoint by itself might be problematic, but if one could couple it with some kind of a clinical outcome.
Right. But we are dealing with antibiotics, and as a first step, if the drug doesn't kill the bug,
sure.
Unless it's got some magical properties like the macrolides do, it's unlikely going to have a significant effect on the outcome of the disease.
But if you do look at clinical outcome in the short term, 6-minute walk test has come up as an example, which again has people, some who like it and some who don't like it. And we've heard from patients this morning as well that their results on the 6-minute walk test could be highly variable depending on what day it's administered and how they were feeling on that particular day. But again, we don't have a lot of good clinical outcome measures here, so maybe 6-minute walk test is a starting point. We have to start somewhere and sort of build upon it. We've heard about the use of FEV1, which might be probably more relevant in a CF population, but I think Dr. Donald mentioned it may not be very helpful in a non-CF patient population. So are there any other clinical endpoints one could think of besides the 6-minute walk test or an FEV1, a pulmonary function test, which is probably more helpful to a CF population? Is there anything else that, from a clinical standpoint, you all have found helpful?
I don't know of anything else existing. I think there's a need to develop more combined disease activity measures, and that's something we should do as we go through all these trials together. And so those would include, objective things as well as subjective measures.
I would say that, in CF where we know the FEV1 is a very important marker to follow, but in NTM, even in CF, we don't know that if you treat them, you reverse that trend of downward with it. So, there are anecdotes, but in terms of even CF where FEV1 has been very useful, in non-CF bronchiectasis, I think FEV1 has not been very useful as a marker of, people come in, the CT's better, the micro's been negative for a year, but their FEV1's worse. And you try to figure out how does that, it just doesn't correlate. So my personal clinical experience is that FEV1 in non-CF bronchiectasis has not correlated well enough that I think it could be used in a trial. Anything more than a secondary outcome.
In our recent look at sort of mortality predictors, it looked almost absolutely useless. We had patients who died with very well preserved FEV1s, and many of the patients came into the follow-up period with very high FEV1s, and it didn't seem to correlate well with mortality at all. So it's not to exclude PFTs in general. There may be other factors, and maybe something like diffusing capacity that might reflect changes in pulmonary vascular involvement that may explain some of the reasons why 6-minute walk may be responsive, may be important. But FEV1 in and of itself does not look like a very good predictor, at least not in our hands.
We need to move on, or we'll never eat dinner.
Right.
Well, there's some other questions, especially under the definitions of sputum conversion, all that kind of stuff, that are, like we said, like Ann said, we haven't.
Right.
It might be we'd all agree on them, but we at least need to bring them up and.
Right. I think it might be more we need to learn more about it, and when we have these trials and we use them as exploratory endpoints, that might be the best way to learn about them. I think in the interest of time, we need to keep moving. If there are no other comments about endpoints, I thought we could spend a few minutes talking about, I skipped number 2 because I think everyone was so eager to get the endpoint discussion, and so I jumped ahead, is if we could talk about use of the controller, what use of controls, whether if they have to be active control or is there any place for placebo-controlled trials. And I think that then ties us back to the patient population, because it might be feasible in a certain subgroup of patients to do a placebo-controlled or a delayed therapy option. So I would welcome your thoughts on controls.
Yeah, I'm glad you went back to number 2, because I think it's really it's really important, and I think it reflects how we answer number 2 or number 3. I might get yelled at by my colleagues. I know I have been before, but I'm a big fan of placebo-controlled trials to show that your drug actually does something. And while we've heard over and over again today that this is not TB, this is not TB, pretty much everything we've heard up here about trial design has been about TB. And we've approached it similarly, and I think TB is very different as well. We're all in agreement on that. You can't really do a monotherapy trial against placebo and TB. Obviously, you can't. Patients with TB have to be treated. It's a public health concern, and then there's also the patient concern. A lot of people with NTM, as Chuck mentioned and Dave seconded, and we've all talked about, don't need to be treated yet. You have this huge pool of NTM. I don't know what proportion it is, but I would suggest it's the majority of patients that when they are incubating their disease for years and years, it takes so long to diagnose. If we could diagnose it earlier, we'd find these people sooner. But, there's a lot of people that don't need to be treated right away, and it's a unique group. It's a unique infection where you could actually do a placebo-controlled monotherapy trial where you take a new drug and you see if it does anything. And if it doesn't do anything, you don't probably need to study it. And it would obviate all our discussions from 3, 4, and 5 because the drug doesn't have in vivo activity and it actually doesn't work. So if it does have activity, then you move to number 3 or 4 and 5 and figure out how to design a trial and how to actually use the drug. So anyway, I'm a big fan of that. You can't do that with TB. You can't do it with most infectious diseases because there's an imperative to treat it right away. So
I thought you had the primate model that we could test this on. Because
we do.
I think you're right. I mean, you hear about people being on quinolones, and you hear about linazolid, and we really, don't have any data, right? Very good data for stuff
I agree, and
done.
The other issue is why haven't we approached this like bronchiectasis? I mean, for at least a subgroup of people with non-CF bronchiectasis, why aren't we thinking about treating the disease or at least studying the therapies in similar fashion? I mean, those are all monotherapy trials. You got people that are infected with something, and you give them something to see what it does to them, or at least does to their bacterial burden. And then there's all the other things to measure. And again, those are different diseases, but the point is you have the luxury of doing it. You can't do that with TB. It's different for all the things that we've talked about. So I put it forward for everyone to criticize, but I think that we should think about doing that with our new drugs as they come out. And I do think we should be doing it in animal models first. We need animal models. We have one. Other people here are working on them. So I do think it's important.
So just a clarifying question, Dr. Winthrop. So when you said it's doing something, you mean in terms of microbiology? Was that the intent?
Yes.
Okay. So were you planning to look at that at an early time point, so within, say, 4 weeks or 8 weeks of initiating treatment, or were you planning to do it much longer, if a placebo-controlled trial is the way you want to go?
I think it depends on how the drug works, and I think it depends on the bug we're talking about. So just as everyone's mentioned, abscessus is probably different than than MAI, et cetera, and certain drugs have different PK parameters, et cetera. So I think you'd have to design the trial with those things in mind.
Just from a practical standpoint, would that be challenging? I mean, to just put somebody on monotherapy for 6-8 months and they have an abscesses? I mean, is that?
Just let me clarify something. This is not for everybody.
Okay, that's what I was
This is not for people with cavitary disease, people who are sick and need to be treated. It's probably not for M. abscessus unless they fit that patient that Dave showed, no progression. And I have non-progressing abscessus patients that are always culture positive, and they're non-progressive. I mean, they don't feel great, but they're non-progressive. So there are those people out there, but I think they're less frequent for abscessus.
Kevin, I think that for clarity, though, this is really I think what you're talking about is an early phase study that would be a relatively short duration and a highly selected population, right? Okay.
So it's something akin to the EBA studies one does for tuberculosis. I think that's what you had in mind.
I just didn't want to say that because I wanted to be different than TB. I really tried hard.
Okay.
But it's not an EBA study. No, I mean, that's very important. This is not 2 weeks. It's 3 months or 4 months. And we don't, acquired mutational resistance is a phenomenon that we know occurs in a couple of drugs, but that's it. So I know there are a lot of unknowns here, but in my mind, this is one of those big areas where we're not talking about TB.
I would ask the question on the opposite end of the spectrum, also understanding all the virtues of placebo-controlled trials, about whether a placebo control is always needed. And if you are starting out with patients with refractory disease, and especially now that we know something about what that target population looks like on placebo, is it mandatory to have a placebo in patients that there's no question that the disease needs to be treated, there's no question that they've failed what they're on, and especially in early phases of putting drugs first in target disease, could it be conceived that you would have a single-arm study looking at efficacy and sort of compare that to historical effects of the natural history of a similar length of time off drug?
Well, as Kevin alluded, I think it would be unethical to have a placebo arm in a cavitary patient.
Yeah, and that was one of the competing concerns about where to set the endpoint in a placebo-controlled trial. So you're competing against the ethical aspects of leaving someone on placebo where they definitely meet criteria to be treated versus having a long enough time to see drug effect. Those two are very dichotomous in terms of where you set that endpoint.
If there are no other thoughts on the control arm, just one question in terms of the optimized background regimen. If one were to go with a trial design where you have a a test drug, you add it to the standard of care, whatever one considers the standard of care, and you compare that with the standard of care, and you have to optimize the background regimen. If you truly end up having a very heterogeneous mix of patients, wouldn't that pose yet another challenge in terms of the background regimen? It would be quite different depending on whom you are treating. So I think that was another reason why we had asked if it makes sense to separate out the patients. So would welcome your thoughts.
Well, failing a regimen is failing a regimen. So if the design is to add the drug onto their failing regimen, with the assumption that there are no other viable options to it, and again, it takes into account whether the drug biologically is thought to be active in the two groups that you're mixing. I'm not sure it makes that big of a difference.
I think the cavitary patients also offer the opportunity of having higher mycobacterial loads, and so it might be a little easier to evaluate efficacy of the drug microbiologically, particularly if you're using a semi-quantitative score.
Yeah, from the industry side, it's a little bit challenging because we would end up having to provide perhaps 3 or 4 different drugs, none of which are approved. So they would therefore all be experimental at that time. So I think just leaving it as optimized background regimen plus or minus the experimental is a much more pragmatic way to go.
And I would just add what Dr. Farley said earlier about the powers of randomization. And I think that, that can sort it out to a degree.
Or you stratify by site or cluster of sites.
Yeah, or even stratify by whether someone has interrupted treatment or not. What we find in the refractory population is that patients are on treatment for a long time and they do take holidays because of the toxicities and it's not working anyway. And so that's an area where you can potentially stratify as well.
If there are no more comments about clinical trial design itself, I thought we could spend a few minutes just talking about some practical difficulties, and I think it had come up for discussion this morning, and Dr. Wallace had made a point about how difficult it is for patients to travel 75, 50, 100 miles if they need to, and maybe, Dr. Eagles, you can provide your perspective as you're actually doing one of these trials.
Yes, the feasibility of these trials is it's very challenging. Primarily because of the length of time you are expecting the patients to stay in the study. Retention is something that you need to work really hard on. And in order to sort of meet the definition of a conversion as it is in the guidelines right now, you do have to have your patients produce sputum every month. Otherwise, how else do you define it? And it is arduous. These centres are not, everywhere, but the patients are everywhere, and so you need to be able to understand and accommodate their travel. So it has a lot of challenges. I think one of the things that we've been told, particularly by the physicians that were in our last study, is that these patients need to just be handheld basically, because it's arduous. They've got the comorbidities, they've got underlying lung disease, they're on all these medications, and I think what we need to do is actually just pay attention to all of that and just help them along.
Dr. Daly asked me to say that monthly visits were not practical.
And I would just reiterate that, and the fact that I don't think the visits need to be monthly. And again, patients can very easily be trained to do standardized collection of sputum and to have that sent directly from wherever they are to wherever the core lab is. So that takes care of your microbiologic outcome. The PROs can be administered remotely. We're not talking about other measurements that are that difficult to do. Things like 6-minute walk or PFTs are fairly standardized by guidelines. And I think if we can think outside the box instead of having everything tightly controlled in a relatively few research centers, and space out the actual visits a bit, we could probably still maintain the frequency of data collection that we would need for the trial.
I mean, you could also arrange to have blood drawn and sent to the centers, and I mean, I think you could do essentially, and this is one place where doing a complete physical exam probably is not, I mean, we fill out the forms, but it really doesn't add much, and probably not totally essential that we do it if you really felt like it needed to be done.
Yeah, I think taking more of a pragmatic trial approach in the future would be useful. I echo.
I mean, it takes them all day to come to clinic.
Sure.
Prepare, come early, blood drawn, whatever needs to be done, sitting there, they miss lunch, and then they're exhausted when they get home. And so by the time they've done that 4 or 5 times, they start thinking, well, gee, I don't want to go today. So I agree with Gina. It's very hard on them, even though they want to be part of the study. It requires a whole day's worth of, when these are people that they're sick, so they get fatigued relatively easily, and they may be down for a week after they make the visit.
Yeah, there's an interesting comment from one of our patients this morning about how they spend 6 months in Florida and 6 months in another state. And so that, to be in a clinical trial and to be geographically that separate is really challenging. So it seems like it is a perfect setting where the PI and the primary care physician have to sort of collaborate and work hand in hand to make this work.
Fortunately, the most common destination is where most of the centres are because of those high evaporative pressures.
Could I ask one last question that of the panel, the serology data that I showed, the IgE, does anybody think that has any value as some kind of marker in a trial?
No.
I think that this is a discussion that's been going on for many, many years of trying to find both a serodiagnosis and a seromarker. It might be something interesting to put in as an exploratory variable.
Yeah, no, I'm joking. I think you should look at it, but I think there probably are seromarkers out there, but that's probably just not the most useful.
If the panel doesn't mind, I just had one last question, and I think we heard in a couple of presentations that the likelihood of reinfection is so high, so 60-70% of patients in fact have reinfection. And the way we are looking to design trials, we are requiring regular visits while on therapy, but certainly a visit at some point after treatment is completed. And, again, as I said earlier, we picked 3 months, but it could be shorter, it could be longer. But then it seems very important at that point one has to differentiate if it's truly a relapse of the infection that they started or if it's truly a reinfection, especially given the numbers are so high. So any thoughts on how we might address that?
I think it's imperative that the isolates be banked. Richard will tell you that's really the only way you can tell is to be able to compare those microbiologic recurrences off treatment to what they started with.
Okay.
So in the last trial, the broth bottles can be saved, so we save every positive that the patient has. So as we said, sometimes there'll be a positive in the middle of 5 negatives, and you want to know if that's the same or not. If you don't have the bottle, then we got to turn it over to you, and you don't know what it is. So really banking those organisms, all of the isolates, even the screen isolates, turn out to be important sometimes when you want to go back and they have a late positive and you want to compare them. So, it's not difficult to do. You just has to be the mindset, and you just set up to do it. And I'd like to think that one of these days most labs will be doing it, because for long-term follow-up, these are long-term patients. It's not like they show up with an E. coli urinary infection, you treat it, and you throw the organism away. These are often patients for life, and they're complicated, and being able to evaluate them, especially in a study. Think of the studies. Think of the British Research Council did that MCANSACI study. They didn't save the isolates, and they had a bunch of relapses, which could easily have been new infections, but they didn't save the isolate, and so the study kind of went down in flames in the sense that they were all above the 5% relapse rate. If you accept all those as being true relapses, it killed them. So.
And so far, I think, when we do see we have a patient that's negative and then comes positive, pretty much parallels the numbers that you had with respect to which ones are a new infection or which ones are relapse. It is a very important question. It means, the difference in the meaning of what each of those is huge. And we did look at it in the last study, and we're definitely looking at it in the next study. I mean, it's very important, but it needs to, after the study, this is within a clinical trial, and it's well and good, but I think it needs to also be information that's available for physicians treating patients in the clinic. And, from what I understand, but I mean, you can tell me, I mean, you're in a great lab, but when we look at some of the patients coming in and they have their history, they have a history of MAC, but sometimes those labs don't even separate intercellulari from avium. And sometimes that's all you need if you're going to have a relapse. I mean, at the very basic level, if you get a different species, you don't have to go any further, but that's not even done.
Okay, so I think we're running out of time. Dr. Farley's reminded me only 3 times that I need to stop talking. So with that, I'll thank all the panel members. I think this was very helpful, and we'll have to take all this back and put our heads together and hopefully design better trials as we move forward. So with that, I'll turn it back to Dr. Farley.
Yeah, this has been great. As with most federal meetings, we will invite a period of open public comment. If you signed up for this earlier and you didn't actually know what you were signing, it's perfectly fine to pass. I'm going to ask Megan to help by using the hand mic. Open public comment speaker number 1 is Dr. Renu Gupta. We would ask you to limit your comments to 3 minutes.
Good afternoon. Thank you very much for giving me the opportunity to speak. My name is Renu Gupta. I am a physician, infectious diseases, and I am working as an independent consultant. I was formerly working with INSMED. And so I just really have a couple of points to make. I would like to thank the agency for holding this forum today. And although NTM lung disease is an orphan disease, it today does not feel like a neglected disease. And this momentum that we've started to gain with all our patients here, our clinicians and researchers and the agency needs to be sustained. I have only a couple of points which probably echo what has been discussed today, nothing novel. One is the unmet need and the seriousness. We need strengthening of our infrastructure within the United States at minimum. And by that I mean if we can have increased outreach to primary care physicians and also pulmonology centers outside the centers of excellence, so that we can have increased awareness around diagnosis of the disease. So this has been discussed for many years, but we now need to put a system in place, an infrastructure. The CF Foundation's registry is a model, and the TDN is a model, and I only illustrate that because I've had the good fortune of working with their team and learning over time as to how successful that has been in terms of outreach for patients, and that clearly had an impact in drawing patients into clinical trials. We have an Ntm Registry, which is a module of the bronchiectasis registry with 13 sites. I've been talking to the stakeholders and making a plea for a stronger public and private sector partnership. I think this is registry, and the benefits of a registry, pre-approval and post-approval, are immense. And the last thing that I would say is related directly to the drugs currently being used and the drugs in development, is that if we could come up with a model of cooperative groups, and like we have in oncology or we have in mycology, and with significant funding and backing from the NIH and some investment from the biopharmaceutical industry, that those cooperative groups could take on the mission of NTM-focused related research agenda. I know the ATS has that in their mission as well. ATS cannot do it alone. And so that would also lend itself to looking at current regimens and evaluating, and I believe there are ways one can do that in a cooperative group studies. And lastly, a plea for my colleagues in the biopharmaceutical industry to go to their management and ask for greater investment in both diagnostics and small molecules and biologics for treating the bacteria and also for improving the underlying inflammatory cascade in the lungs of these patients. So thank you very much.
Miss Mary Fisher, could you identify yourself? It's right here.
I just want to make a comment regarding clothosamine. As I alluded to it when I was on the panel, I started clothosamine back in 2011. For those of you who don't know clothosamine, it was a medication that was used to treat Hansen's disease or leprosy. It no longer is available commercially, and when they prescribed it for me, my physician had to fill out reams of paper, jump through 1,000 hoops just to get it approved. Once it was approved, then my physician had to order it from the pharmacy in Louisiana. It had to be delivered to the pharmacy or to the physician office, and then I had to drive an hour to go get it. My concern is that medication is an old medication. I understand when they started using it, it was investigational. However, I've been on it for 4 years, and I'm sure there's quite a few people in here that have probably been on it or are on it. I don't have any side effects. The one side effect that is very common that I would have loved to have had was the bronze skin. I'd love to have a tan year-round, but I never got it. Anyway, what I am requesting is if we cannot streamline that process of getting that medication to us, I have to still call my physician's office, and then when they went to request it again this year, the process changed. In January, they decided that they no longer were going to pay the postage, which I have no problem with. However, that was not communicated to my physician's office until April. When I went to get it renewed, they tried to reorder it the old way. They told them it wasn't the proper way. The incompetence in the doctor's office didn't get it done, and I was a week without the medication after many, many phone calls to the office to remind them to get the drug. Now, I'm responsible for generating the postage. I'm responsible for sending the email with the postage, but it still goes to the doctor's office, and I have to go pick it up. I don't understand why that can't come directly to me. If I'm doing all the work, I'm doing all the generation, why is it that I have to still jump through the hoop? And I can guarantee you my physician doesn't even know when it arrives. It gets to the receptionist, they call me, I pick it up. So I am just asking for them to look at that and see if that drug cannot come off that investigational and streamline the process for us to get that medication.
Thanks. And just so people don't say something in a vacuum, so we might have talked to you on the phone a bunch of times because we actually have about 3 full-time equivalents working on the clothosamine expanded access program. So we have heard you loud and clear, and we're working very hard to streamline it. Part of it is that because it's not marketed in the United States, it is only available through the single patient IND program, and those regulations, unfortunately, require us to deliver it to the physician, not to the patient, even though the patient paid us to deliver it to the physician. So, but we are working hard on streamlining, and it's a priority here at the FDA to get that done. So, be patient, and we appreciate your patience so far. Part of it is that through that process, we actually have gotten to know a fair number of the patients, and we find that really helpful because we get to talk to you and we know kind of more what your life is like and we're more connected to what you're going through. But there might be a better way to do that. So we hear you. Next is Marcy Wayne, or W-E-I-N-E? Weiner. Sorry,
I have three things. The first would be early diagnosis, perhaps through a skin test, such as they do for TB. And is that a possibility, and is anybody working on that? It seems to me it should be feasible. I just don't know. The second issue is something that Dr. Gupta brought up, which is the inflammatory cascade. I'm very well aware of that, and oftentimes when I try to bring in some sputum, I'm really getting what looks like to me to be inflammatory. You know, it's just white. It's white and cloudy, but it's just really inflammatory and not necessarily bacterial. And I'm not, as a patient, I say, okay, maybe I should try antihistamines or this or that. What about low-dose aspirin? But I don't hear anybody really, as far as the the pharmaceuticals, trying to deal with this cascade in the lungs, of the inflammatory in the lungs. And I think that's very critical, especially with us with bronchiectasis. I think it's perhaps the number one thing that we need to do for bronchiectasis patients. And third, I know it's an old one. I think it has some eucalyptus in it, which is a known antibacterial, was used in the Vicks Vapor Rub. And those of us that are old enough remember our mothers putting this in a vaporizer, rubbing it on our chest. But it also helped to break things up and brought relief. And perhaps in a more modern form, in delivering and through either an inhaled or nebulized form, eucalyptus could be used. I mean, this has such strong antibacterial properties. I just can't believe that we're not utilizing this in any fashion. Now, I say this as a very person who did microbiology so many years ago. I'm probably useless for you, but I just can't think that's a thought to spark some kind of look into. It's very easily accessible. They have a lot of it in Australia. I brought some back. And in California, and you can get it in Arizona. You go to a spa in Arizona, they're going to have little bottles of it. And for the ladies with bronchiectasis that are told they cannot take showers, but they have to take baths, put 5 or 6 drops, little drops of that in, and get the benefit of it. But I mean, I think for scientific purposes, it would be good to have something looked into.
Miss Deborah Schwartz.
Very quick question. I just wondered how many people here have cavitary disease. If anyone has had surgery and recurrence after surgery.
Did you want folks to raise their hand?
I mean, I can tell you it happens. Remember that surgery is a debulking procedure. It does not... did you have it on both sides or on the same side?
Upper right and middle left by Dr. Mitchell. And for 4 years I had negative sputums. I had one positive in February. Dr. Daly and my doctors decided I should go back on medication, and I did, but I've never had another positive sputum, and they still want me to stay on it for 18 months. I guess they're worried about cavitary disease again. I don't know. But anyway, I'm taking the medication. I'm doing okay. Inhaled amikacin I like very much, but I only had one positive 4 years after surgery.
I'll just say that our experience from fingerprinting is that people who have relapse with the same strain almost all do so within 6 months. I've never seen a relapse with the same strain after a year. So almost certainly your isolate is a different isolate.
It is a different one. It's intercellulary.
And so on the basis of a single, I can understand why they're concerned, but the answer is we would not put you back on medicine for a single positive. We just collect samples and follow you. I mean, I don't know that there's a right answer here, but that would be my approach.
Okay, great. Well, thanks very much, everyone. So before I forget, I need to announce there's a shuttle bus. Phil, are you involved with the shuttle bus? You're not. Okay. But there is a shuttle bus that some of you came on. Right. It's leaving from the circle out front at 5:15. So we're around. If you have trouble finding the way you came back in, let us know and we'll get you headed in the right direction. I want to thank everyone, particularly the patients who I know have traveled. Many of you have traveled a very long way. I was told at lunch by the patient-focused drug development group that this is one of the best meetings we've had in terms of the quality of feedback that we've gotten from patients. This has been extremely helpful to us. I can tell you from early on in the HIV epidemic that the progress that got made because patients and academics and developers of drugs and regulators were talking on a regular basis and knew each other and working together. And I'm seeing that happening in MTM, and I'm very excited about that. You've been a big part of that process. I want to thank the panel in particular for taking time out of your schedule. This has been very valuable to the division as they begin to work on clinical trials. I know that there are some companies here who are thinking of working in this area, and we want to encourage you, and we also want to remind you that the division is available to meet with you at any point in your program, particularly early under the pre-IND program, and provide you regulatory advice. And that's a fairly easy thing to access. If you don't know how, just look up Samadhi or I, and we'll get you pointed to the right person. So with that, I want to thank everybody for a great meeting, and we look forward to seeing many of you again soon and talking further as we begin to make progress, continue to make progress in this area. So thanks very much, and safe travels, everyone.